Welcome. We are now in paper 3 from Maktab Rendah Sains MARA. So a typical paper 3 in SPM Biology, of course, you are expected to have two questions. Question 1 is 33 marks and question 2 is 17 marks. Although each of the question carries a lot of marks, the good news is the questions in paper 3 are very structured and they are also very modular, which means that even regardless of the year the SPM question is coming out, they usually follow the exact same format. So you can more or less predict and anticipate the kind of or the style of the question that will be asked. So let us go to the first question, which has the most marks. So normally in the first question you will be given a statement or a diagram, and then you will be asked in many subsections various types of questions. So here we have a statement talking about a fresh fish fillet, and then by rinsing it with tap water it will cause a lot of problem because of this osmosis process. However, the most important thing that you should focus your attention to is usually at the lower part here. So here you are requested to determine the concentration of solution that is isotonic to the mackerel fish fillet, and so you have to look at the steps that are being carried out. So here will be the step 1, where they have 3 different fish fillets that were wiped with tissue paper, weighed, and then they record the initial mass. So here you have to take note of the initial mass, and this would be 50 grams. You don't have to write it down in the answers, but this you have to take note. Step 2, the fish fillet is immersed in a beaker that contains 200 ml of 0.1 molar sodium chloride solution. After 30 minutes, the fillet was removed from the beaker and wiped again, and then afterwards the final mass for each of the fish fillet were weighed and then recorded. And then they repeated this entire experiment, now using 0.4 molar of sodium chloride and 0.6 molar of sodium chloride. And then the results are being tabulated. So this is a very typical question 1, paper 3, and you will be given various type of scales and various types of diagrams whereby you have to read out. So here you are supposed to record all the different readings. Remember, when you have to do this, always include the unit. So unit is of utmost importance. So if you put the number without the unit, then you will not be able to get the full marks. So here is 70 grams, this is 74 grams, this is 46 grams, this is 44 grams, and this is 42 grams and 40 grams. So this is the first part. Record the final mass of the fish fillet in the boxes, and which we already did. And for this, if you get everything correct with the unit and all, you should be able to get the full 3 marks. Next comes to the observation. So normally in this type of question 1, you will be asked to give 2 observations. There are some tricks to do this. So first of all, you must always refer to the values that comes from the manipulated and also the responding variable in the table. And remember, this is an observation, so you just write what you see. You do not have to explain anything at all. This is not the section for explanation. Just write what you see and use the exact values that you can see from the tables above. And normally, because you have 2 observations, you can put usually the observation that has the highest value of the responding variable and also the lowest value for the responding variable. So in this case, let us go up to the diagram here. Here you have 70 grams, 74 grams. This will be the highest value coming from 0.1 molar sodium chloride solution, and the lowest would be coming from 0.6 molar sodium chloride solution. You have 42 grams and 40 grams. So you can write both of them. How do we do it? So you can write something like this. At the concentration of 0.1 molar sodium chloride solution, the final mass of the fish fillet is 70 grams in the first reading and 74 grams in the second reading. So over here, you state a few things. So first of all, you state that this is your manipulated variable over here, and then the final mass of the fish fillet is the responding variable, followed by the exact value that is shown in the diagram. Okay, similarly, for the second observation, you can say at the concentration of 0.6 molar sodium chloride, final mass fish fillet is 42 and 40. So here again you have your manipulated variable, the sodium chloride concentration, and your responding variable, which is the final mass of the fish fillet, followed by the exact values that you see. Next, after you give your have given your observation, you will be required to give two inferences based on the observation. So the format that I will suggest is as follows. First of all, you need to remember inference means that you have to give a reasons. The format could be something like this. The observation 1 is, what is the condition, because of, and then give of a reason. To illustrate what I meant, so you can start something like this. So this is based on observation 1. The final mass of the fish fillet in 0.1 molar sodium chloride solution, which is what you saw above, is what is the condition? Is the highest, because then you need to provide suitable solution based on the fact. So because of 0.1 molar sodium chloride solution is hypotonic, and therefore water is able to diffuse into the fish fillet cell by osmosis. So this one here in orange, this is the reason, which is based on a well-established fact. So osmosis, water diffusion, and also hypertonic solution. Similarly, in observation 2, you can put it something like this. The observation 2 here, which is 0.6 molar sodium chloride, is, what is the condition? Lowest, because 0.6 molar of sodium chloride is hypertonic, and therefore water diffuses out of the fish fillet cells by osmosis. So if you follow this standard format of giving reasons based on the observation, you should be able to answer this section without much of a problem. Always relate it back to observation 1 and observation 2. Next, you will be required to state the 3 different variables for this experiment. So the first variable that you need to put is the manipulated variable, followed by the responding variable, and you also need to state what is the constant variable. Manipulated variable, it means that what has changed in the experiment. As for the responding variables, it means what are the changes that follows after you change your manipulated variable, and constant variable means what is it that is not changed throughout the experiment. The manipulated variable in this experiment would be the concentration of the sodium chloride, because 3 different concentration at 0.1 molar, 0.4 molar, and also 0.6 molar were used in this experiment. So this shows very clearly that the concentration of the sodium chloride solution is the manipulated variable. As for the responding variable, you have the final mass of the fish fillet, also the average mass of the fish fillet, and the difference in the mass of the fish fillet. So basically all the mass of the fish fillet, different aspects of it, are considered the responding variable. And finally, for the constant variable, you can put in the type of fish. So the same type of fish that is being used would be the constant variable. So filling in the different variables usually is not so much of a challenge. However, the methods to handle the variable could be a little bit tricky for some of the students. So here the method requires you to put in a few keywords, usually are verbs, and also, like for example here you have to say use different, and so the keyword here different is very important to show that the variable is being manipulated. So here you use different concentration of sodium chloride solution. As for the responding variable, normally you need to put a few keywords as well. So here will be in green, weigh and record, as well as calculate. So these are the 3 words which are really important. So first of all, you need to put weigh and record the final mass of the fish fillet by using a balance, and then you also need to say how you calculate this. So you need to say calculate and record the average final mass of the fish fillet by using the following formula. Please show the formula as well. So how do you get an average? So final mass 1 plus final mass 2 divided by 2. And also say how do you find the difference in the mesh of the fish fillet. So this would be by putting calculate and record the difference and then using this formula. So average of final mass minus the initial mass. So this would be the method. So it's alright to put more just to be sure. And finally, the constant variable, you can say something like fix the same type of fish. In this case is the mackerel. So the keyword will be the fix and also same. So remember all these keywords when you are writing for the method to handle the variable. Next, once you have finished the variable section, you will be required to state the hypothesis. So when it comes to stating the hypothesis, it means that you are supposed to predict and also state what is the expected outcome of this experiment. In this aspect, it doesn't matter if you have predicted wrongly or you expect it wrongly. That's okay, because it's a hypothesis and you are supposed to test it out. And more importantly, you must always relate the manipulated and the responding variables in this statement. So for example, here you can say in the following format, as the concentration of sodium chloride solution, so this will be your manipulated variable, increases or decreases, the final mass of the fish fillet, this one here is your responding variable, decreases or increases. So here you could see there are two elements in this one sentence. First of all, you could see that you have a manipulated variable and a responding variable, and the relationship between them here, increases, decreases, decreases, increases. So it's inversely related to each other. So remember, for hypothesis, you must state a predicted outcome and always relate the manipulated to the responding variable. Next, you are usually asked to construct a table to record all the data from the experiment, and usually they will provide you with the titles that needs to be in the table. So when you are provided the following titles, make sure to include all of them in your table. Here you are supposed to put in the concentration of sodium chloride solution, the initial mass, final mass, average final mass, different in mass. So if you look at all of this, they can be broadly categorized into two things. So one here would be your manipulated variable, and the rest will be somehow related to your responding variable. Manipulated variable will usually be placed on the left, and the rest of it will be placed on the right. Over here, this is how a typical table will look like. You have the concentration of your sodium chloride, which is 0.1, 0.4, 0.6, and the mass of your fish fillet consists of the following sections. So initial mass you need to know from the beginning, so this is 50 grams. Final mass, the first and second, this is what you recorded, so you transfer all the data here. And the average, so you need to calculate, put it in, and you also need to put one section here that is related to the change in the mass of fish fillet. The three important criteria for the 3 marks that is allocated for this section is: number 1, you must have all of the titles; number 2, you need to record all of the data correctly; and then the third one, usually you need to put the calculation. If you have all of these 3 correctly done, you should be able to have the full 3 marks. So once you have constructed the table, the next part usually will ask you to construct a graph on the graph paper that is provided in the question paper. So here you need to pay careful attention on the type of graph, and also what are the aspects that you need to plot onto the graph. So here they ask you just to draw a graph, and then you need to show between the difference in the mass of fish against concentration of sodium chloride. So this means you need to plot here the concentration of sodium chloride against the difference in mass of fish, or the change in mass of fish, so this one here, and not the average. So these are the two data that needs to be plotted onto the graph. So let's move to the graph first. Alright, so this is how the graph will look like. If the axes are not already labeled, make sure that you label the axis. To recall, the x-axis will be the place where you put in your manipulated variable, and the y-axis will always be your responding variable. So in this case, the concentration of sodium chloride is your manipulated variable, so it's in the x-axis. The difference in the mass of fish is the responding variable, so it's in the y-axis. And then you make sure that you transfer all the data points and then draw a line. So this is also 3 marks, and the 3 important criteria for this kind of graph will be, the first one is that you use constant scales on both axes. So make sure this and this axis has a constant scale, so they are regularly spaced 0.1, 0.2, and not something like 0.1, 0.5, 0.9. So there's no constant interval in between them. So make sure you have a constant scale. Number 2, you transfer all the points correctly, so ensure and double check that you have the right values for both x and y axis. And the third, make sure you draw a line, or in some cases, if they ask you to draw a bar graph, to make sure that all the bar graphs are properly connected. Okay, so this would give you 3 marks. And then the next section here, after you have drawn the graph, usually they will ask you to predict something based on the graph. So here it says, based on the graph, you need to identify the concentration of sodium chloride that is isotonic to the fish fillet. So when it means isotonic, means that then there should be equal rate of diffusion of water in and out of the cell, which means there should be 0 change in the mass here. So this is the point where the line cuts and it is at the 0 change. So this is a change in mass, so there should be 0 here, and this would be the point, the isotonic point. So here would be approximately 0.33 plus minus 0.1 molar. So you can state number 1. So you state it here. Isotonic concentration is 0.33 molar plus minus 0.1 molar. This would be 1 point. And you need to also explain why is that so. So this is because the rate of osmosis is 0 gram per minute, which means there's no change in the mass due to the equal diffusion of water molecules in and out of the fish cell. Right. So the next part is usually a different experiment, but somehow related to the current question. So the group of students carried out this experiment where they put in 500 grams of a live mackerel fish into a seawater inside an aquarium. So you are required to predict what happened to the final mass of this mackerel fish. One very important trick, or one very important point here, is that the fish is covered by skin and also it has scales. So these two actually ensures that there is no osmosis that will occur, because the skin and scale they are impermeable to water molecules. So if you were to predict, in this case, it means there should be no change in the mass because there is no osmosis. The way you write it, you are also here, you are allocated 3 marks. So first you need to state whether the mass changes or not. So it goes something like this. The final mass of mackerel fish is equal to 500 gram because the skin and the scale is impermeable towards water. And because of that, there is no osmosis process that occurs, because no water molecules is able to move in and out of the fish cells. So you need to state and provide the explanation of your prediction. And one of the most challenging section in paper 3, which is to provide an operational definition. So when it says operational definition, it means that the definition has to be based on the experiment that is being conducted. In this case, you are required to provide a operational definition for osmosis. So you can say something like this. So osmosis is a process of movement of water molecules into the cells of the mackerel fish fillet. So here in blue, this is directly related to the experiment, as shown by the average final mass change in the mass of fish fillet affected by the concentration of sodium chloride solution. So if you see all the points in blue, they are directly related to the experiment. And so first point you need to write is, of course, the definition itself. Secondly, how this was determined. So normally this is coming from your responding variable. So this is the average final mass change in this case. And then thirdly, you need to say that the mass of this fish fillet is affected by what? So it's affected by your manipulated variable. In this case, it will be the concentration of the sodium chloride solution. So these are the 3 points that you need to put in. So first, the definition. Second, how you get it. And third, the relationship between the manipulated variable and also the responding variable. And finally, the last section here for this experiment. So here it shows that in another experiment that the water spinach was immersed in a 0.8 sucrose solution. after 1 hour, this is what happens. So if you see, this is the start, this is the end, no change has occurred in the shape, this water spinach. So what does it mean? It means that the solution would then be isotonic to this water spinach because there is no change in the shape. 0.8% would be the isotonic point. Now they repeated this experiment using different concentration of sucrose solution, and you are required to classify the various concentration of sucrose solution into either hypertonic or hypertonic. So hypertonic will be anything that is less than 0.8 because this is the isotonic point, and hypertonic will be anything that is more than 0.8%. So now it becomes very easy. So 0.3, 0.5, and the rest of them 0.9 to 3, they all belongs to the hypertonic.